evolution of life stages. If you go from the most unevolved to evolved stage, what do you want? If I give you 4 things to arrange: eukaryotes, or bacteria, prions, viruses, viroids. Arrange these 4 in the order of evolution. Which one is the least evolved? Prions. What are prions? Prions are infective proteins. That's all. Then next is viroids. Viroids are naked genetic material. Naked genetic material. Alright? Viruses are genetic material surrounded with protein coat or a sheet. Then bacteria, prokaryotes. If you have to pick the odd one out among these 4, which one do you pick the odd one? Pick as the odd one. Prions, viroids, viruses, bacteria. Which of these 4 is the odd one? Huh? Why? Because it is protein. Viruses also have protein. Bacteria also has protein. It doesn't have RNA. Viruses may not have RNA also. What if it is a DNA virus? Huh? It still has nucleic acid. Okay. Viroids, viruses and bacteria, all 3 have nucleic acids. Prions does not have nucleic acid. Okay. Another perspective. Give me one more perspective. Yes, prion can be an odd one. Then one more. What else can be the odd one? There is one more possibility. Come on, quickly. We should finish this paper today. One paper, one day. Bacteria, why?
It's the most advanced.
It's the most advanced, okay. Logic refinement required. Bacteria is correct, but it is not the logic. Come on, quickly.
Cellular.
Huh?
Bacteria is cellular, sir.
Bacteria is cellular, okay. More logic. Bacteria is right. More logic. Bacteria is independently living. The other 3 are not living. Viruses can become functional only inside a host. Viruses are obligatorily parasitic. The other 2 are non-living, not living. They are just chemical components. Alright, anyways. So what is the right choice here? Viroids differ from viruses in having RNA molecules without protein coat. And viroids always RNA. You don't consider naked DNA as viroid. Why RNA is capable of producing disease? Why not a naked DNA fragment? That is not known. How it actually does it? Not known. But viroids are naked RNA without a protein coat. Anaphase promoting complex is a protein degradation machinery necessary for proper mitosis of animal cells. If APC is defective in a human cell, which of the following is expected to occur? See, anaphase promoting complex. You might not have studied it earlier. That's fine. The question itself is giving you knowledge. Anaphase promoting complex will promote what? What phase of mitosis? It will promote anaphase of mitosis. What happens during anaphase? The chromosomes which are aligned at the equator gets separated. The chromatids get separated so that the chromosomes move to the opposite poles. If something that promotes anaphase is defective, what will not happen? This separation will not happen. That's all, no? So look at the options. Chromosomes will be fragmented. Is the fragmentation of chromosomes that anaphase is doing? No. Chromosomes will not segregate. Recombination of chromosome arms will occur. Chromosomes will not condense. Anaphase is not related to any of those. Anaphase is related to separation of chromosomes or segregation of chromosomes. So if anaphase promoting complex is not present, what is supposed to happen in anaphase will not happen. So, chromosome will not segregate is the right choice. Chromosomes will not condense. If chromosomes do not condense, defect is in which phase of mitosis? If chromosomes do not condense, defect is in which phase of mitosis? Prophase. Because condensation of chromosome is happening in prophase. If chromosomes do not decondense, if decondensation of chromosome does not happen, where is the defect? Telophase. Telophase. If chromosomes do not align at the equator, where is the defect? Metaphase. That's all. So work in a reverse order. If something is promoting something, so what is supposed to happen? If this is defective, that will not happen. During DNA replication, Okazaki fragments are used to elongate. Don't look at the options. Think of DNA replication. What is formed during DNA replication? What is this? If these are the two strands of DNA, this is replication fork. Replication fork. The direction in which the new strand is produced. Direction always, every time questions related to this comes, I will always ask you the same things. The same fundamentals are always very, very limited. You stick to that, you will not go wrong. What is the direction of DNA replication? 5' to 3'. At the 5' end, what do you have? What is available at the 5' end? Phosphate group is available. Open phosphate group or available phosphate group is 5'. At the 3' end, what is available? Available hydroxide group or OH group. Now 5 to 3 is the direction of synthesis of the new strand. The strand in which is the strand against which continuous replication occurs. Tell me the polarity of the strand of DNA complementary to which continuous replication happens. 3 to 5. Very good. Because the new strand 5 to 3 goes on continuously. So its complementary strand is 3 to 5. Okazaki fragment is formed complementary to the strand that runs in the direction 5 to 3. That runs in the direction 5 to 3. So now this is the replication fork. Let us say my left hand is 5 to 3. So my left hand is 5 here, goes on like this 3. My right hand is 3 to 5. Against which hand or complementary to which hand will the leading strand be formed? Left hand or right hand? Right hand. Against this, leading strand is formed. The new strand synthesis will be from the periphery to the replication fork. On which side? Periphery towards the replication fork is the strand that is leading strand, because the template is 3 to 5. So the new strand synthesis will happen from the periphery. 5 to 3. It starts moving towards the replication fork. But look at this one. This is 5' end. This is 3' end of the fork. This is the fork. So new strand synthesis should begin at the 5' end. This much is only open. Synthesis will start from here. And it moves away from the fork. Moves away from the fork. Isn't it? Now look at the options. During DNA replication, Okazaki fragments are used to elongate. Are Okazaki fragments related to leading strand or lagging strand? Lagging strand. So remove the options which has leading strand. Don't even read that. Lagging strand towards the replication fork. Lagging strand away from the replication fork. What is the right choice? Lagging strand away from the replication fork. That's all. Okay. So whenever you look at the question, try to gather some knowledge that you already have in your mind. Get a picture of that. Visualize that. Then look at the options. It will give you clarity. Genotype of a husband and wife are IA, IB and IAI. among the blood types of their children, how many different genotypes and phenotypes are possible? Don't look at the options. Just write down the genotypes and phenotypes. Just write down quickly. It will take you 10 seconds to write it down. If you don't write it and try to calculate in mind, it will take you more time. Put it down on paper. Just quickly write down and tell me the number of genotypes and phenotypes possible. Where you have to put together calculations, combinations. Writing down is quicker than trying to think in your mind. How many? 4 genotypes and 3 phenotypes. 4 genotypes are IA, IA small I, IA, IB, IA and IB small I. 4 genotypes. Phenotypes are AB and AB. That's all. Over. There is one more antigen that is present on the RBC associated with the ABO system. Which antigen is that? Associated with ABO system. Come on, we have discussed this. That is the precursor protein to A and B. Presence of this protein is by default. The ABO system, gene I, will give rise to this protein irrespective of whether the allele is IA, IB or small I. If IA is present, this protein will also get converted into A antigen. If IB is present, this protein will also get converted into B antigen. If small I is present, it remains as it is. Which antigen is this? Which protein is this? Be louder. H protein or H antigen. Why is it significant? Why is H antigen significant? Bombay blood group. Bombay blood group. What is the speciality of ABO blood group system? Absence of an antigen means the immune system is capable of producing antibody by default. Absence of antigen is by default antibody production. So if H antigen is present in all blood groups A, B, AB and O. So normally you don't produce antibodies against H antigen. But Bombay blood group is that where H antigen is absent. If H antigen is absent, can the person's RBC produce A antigen or B antigen? Can it produce A antigen or B antigen? Just a moment. Hello. Maharaj Namaskara. ಮಾತಾಡ್ಬೋದು ನಾನು ಕ್ಲಾಸಲ್ಲಿದ್ದೆ ನಾನು ಶಿಲಾಂಗಲ್ಲಿದ್ದೀನಿ ಮಹಾರಾಜ್ ಮತ್ತೆ ನಾನು ನಿಮಗೆ 12:30 ಮಾಡಲ ಇಫ್ ಇಟ್ಸ್ ನಾಟ್ ಅರ್ಜೆಂಟ್ ಓಕೆ ನೋ ಪ್ರಾಬ್ಲಮ್ ಎಸ್ ಮಹಾರಾಜ್ ಐ ಆಮ್ ಅವೈಲೇಬಲ್ ನಾನು ಹೇಳ್ಬೋದಲ್ಲ Yes, Maharaj. Thank you. No. See, with ABO system, what do we have to remember? Absence of antigen is antibody production ability by default. Our immune system otherwise will always work on the principle of exposure to antigen, antibody production. But here in ABO system, absence of antigen, you produce antibody by default. No need of exposure. H antigen you always have, so your body does not produce antibody to H antigen. But without H antigen, A and B cannot be produced. That means somebody with defective H antigen, even though their blood group might be IA, IB, doesn't matter, it will not produce A and B antigens. And their body is capable of producing what? Antibody against H also, apart from A and B, because all three antigens are absent. Now when you check for the blood group of this person, generally bedside blood grouping does not involve checking for the presence of H antigen. We will check for A and B antigens. If A and B are absent, what do we consider that to be? What do we consider that? We consider that as blood group O. You transfuse a person with blood group O when he is requiring blood transfusion. That O group will have what antigen? The O blood group will have H antigen. Now this person recipient, he does not have H antigen. You are identifying it as O blood group. You are giving O blood group to this person. Exposure to H antigen, what will his immune system do? Produce massive antibodies against the H antigen. And this is blood, no? RBCs you are giving. So there is enough time for the body to identify that antigen and produce antibody. Unlike the pathogens, which can be removed also. By the time the antibody is produced, the pathogen can be removed. But these blood groups, blood cells will be there. So you produce antibodies and attack the RBCs with H antigen. Massive blood clotting can lead to death also. Remember this, Bombay blood group. Questions have come earlier. I think in one of the previous papers we had a question on Bombay blood group. No? I have discussed this during this discussion only. So H antigen, A antigen, B antigen. Absence of H antigen, Bombay blood group. Antibody absence, default production of antigen absent, default production of antibody. Okay. Is it the same case with RH blood group? Is it the same case with RH antigen? Absence of antigen, default antibody production? Is it so? No. RH requires exposure to antigen for antibody production. Tell me the condition in which there is RH exposure leading to some problem. Erythroblastosis fetalis. Erythroblastosis fetalis is also referred to as HDN. What is HDN? Hemolytic disease of the newborn. Hemolytic disease of the newborn. Can it be treated? Treatment is by blood transfusion. Can it be prevented? Preventing a disease in a baby by treating the mother. Correct, no? The disease can happen in the baby. Treatment has to be given to the mother. When? See, this is a unique treatment, no? You are treating the mother today for a baby that might be born a few years later. To prevent a disease that can happen in a baby that is born a few years later. Okay, next. Which of the following RNAs should be the most abundant RNA in animal cells? Which one is most abundant? Which RNA is most abundant? Think about it logically. Alright? mRNA is the recipe. mRNA is the recipe. tRNA is the grocery. rRNA is the machinery. Or the people who cook. Alright? mRNA is the recipe. tRNA is the one which will bring the grocery. rRNA is the one which will manufacture the food, which will prepare the food. In a restaurant setting, which one should be more? Those who make the recipe, or those who bring the grocery, or those who cook? Those who cook have to be more, no? Similarly, rRNA. 80% of the RNA is rRNA. Least in terms of its concentration? Which one is least in terms of its concentration? mRNA. mRNA, it is the recipe. You don't need too many of it. One recipe, some people to bring grocery and lot of people to cook. Lots of people to cook. Spliceosomes are not found in cells of fungi, animals, bacteria, plants. Now let us assume you don't know what spliceosomes are. Looking at the options, can you make some sense of the options? There are 4 organisms given. Can you make some sense of the 4 organisms? Can you give me some insight into the 4 options? One is prokaryote, other 3 are eukaryotes. One is prokaryote and the other 3 are eukaryotes. So if there is a difference, something has something and the others don't have something, is there a possibility that you can, if you have to take a chance, you can pick the odd one, because the question is, something is not found in somebody. One is prokaryote, other 3 are eukaryotes. Now if this something is present in the one which is not the odd one in terms of its existence, then it should be something that is present in both prokaryotes as well as eukaryotes. So if you don't know what this thing is, your guess should be, which one do you mark as a guess? Fungi, animals, bacteria, plants. Which one do you mark it by guesswork? Come on, put some logic and make a guess. Bacteria, and that is the right choice. Okay. mRNA is also called cistron. mRNA is also called cistron. Cistron is the copy of the genetic code from the DNA. Now this cistron might have actual portion which will code for a protein and those non-coding regions. There might be coding regions and non-coding regions. Coding regions in a cistron are referred to as exons. Non-coding regions are referred to as introns. Spliceosomes are those which will help in the maturation of the mRNA by chopping off the introns. mRNA is copy of the DNA. Before it starts translating, you have to remove the non-coding portions. And when you remove the non-coding portions, all the exons are lined up, which can be translated. Chopping off of the introns is done by spliceosomes. That's all. They remove the non-coding regions. Alright? Bacteria do not have introns at all. The mRNA of bacteria does not have introns. They only have coding regions, no non-coding regions. That means no junk DNA. There is no junk DNA in the mRNA of bacteria. Makes sense because bacteria are the... See, why does junk DNA accumulate in a cell? We have loads of junk DNA. Lot of our DNA is junk DNA. More than 99% of our DNA does not code for anything, which we can relate to now. Why do we accumulate junk DNA? We have a very long evolutionary history. Yes or no? From the unicellular organisms that started evolving, that started appearing on the planet, to the stage where we are, there is continuity of DNA transmission, there is continuity of genetic transmission. But because of evolution, we are not expressing all the genes that we had throughout our at different stages in our evolutionary history. Now all those DNA fragments which probably would have been expressible genes at some point in our past is not useful today, becomes the junk DNA. It does not, no longer gets translated. It is there. Bacteria is the simplest of life forms. It is a unicellular organism, simplest of life forms. Its evolution has not taken it through various stages and it has not evolved beyond. It is still prokaryotic. So there is very minimal DNA which is not usable by the bacteria. It has all exons therefore. Alright? If there are 999 bases in an RNA that code for a protein with 333 amino acids and the base at position 901 is deleted such that the length of the RNA becomes 998, how many codons will be altered? 999 base pairs are there, so it codes for how many amino acids? 333 amino acids. Each amino acid is coded by a triplet code. Up to 900 it is normal. 901st base pair is deleted. 901st base pair is deleted. So how many normal amino acids will be produced? Up to 900? 300 normal amino acids are produced, and beyond that, from the 301st amino acid, there will be alteration. So how many amino acids will be produced now with alteration? How many codons will be altered? 33 codons will be altered. The last codon will not even be a triplet. Correct? Now let us say, I'll change the question. Same question, but the condition alteration is, the deletion also is 901. Assuming that the last codon, assuming that the code does not encounter a terminator codon, how many defective amino acids are likely to be produced? Assuming that the code does not encounter a stop codon at any point, how many defective amino acids will be produced in this particular case? 32. Why? 901st base pair is deleted, so 98 base pairs are remaining from the point of deletion. 98 base pairs will give rise to 32 amino acids with 2 base pairs left out at the end which will not code for any amino acids. And these 32 will be altered sequences because of deletion, frame shift mutation. Those 32 are likely to be defective. Why do I say likely to be defective and why not certainly defective? When the frame shifts, it is not necessary that every amino acid will be defective. The shift might lead to a particular changed codon which might still give rise to the same amino acid which was supposed to be given, which was supposed to come at that particular point, because of the property of degeneracy of genetic code. One amino acid can be coded by more than one codon. Alright? Are you understanding it? So question can be anything, think about it logically and you will be able to answer that. Among the following characters, which one was not considered by Mendel in his experiments on pea? Very easy. Which one? Trichomes. Trichomes were not studied by Mendel. All the other three, the seed, color, pod, shape, stem, all these were considered. Green seed is dominant or recessive? Green seed. Seed color and pod color, they are opposite for dominant. Why do you need roots coming and getting exposed to air? Under what conditions you might need such kind of adaptation? Do we need such kind of an adaptation in aquatic plants? Do we need such kind of an adaptation in aquatic plants? The roots want to breathe, so they come out. They are aquatic plants. They are adapted to take oxygen from the water. So roots which have the need to come out of the soil for breathing should be those which do not have aquatic adaptation. They are not adapted to the aquatic environment. They are terrestrial plants. But why do they have to come out? There is space in the soil also, no? Why do other plants don't bring out their roots? The soil is aerated. There is air pockets in the soil also. Where is it possible where soil does not have airspace? Waterlogged soils. It is not water. Soil is always having more water, so the spaces are filled with water. Mangroves. Alright? Mangroves or marshy areas. Which kind of plants are those which grow in such conditions among the given 4 options? Halophytes, samophytes, hydrophytes, mesophytes. Halophytes. Halophytes are those which are adapted to surviving in marshy areas. Okay? Saline soils and marshes. What are samophytes? Samophytes. Desert plants. Dry areas. Plants that are adapted to sandy deserts. Dry areas. Hydrophytes are those which are adapted to water. Mesophytes are our regular plants which grow in normal, moderate conditions. Alright. What adaptation you might find in hydrophytes? Root adaptations. Floating roots. You might find floating roots which are surviving in water also and can get exposed to air as well. The association of histone with a nucleosome indicates what? Association of histone with a nucleosome indicates what? What is a nucleosome? See, different stages in the maturation of DNA to become the genetic material. DNA, you coil the DNA around. What do you coil it around? Histone proteins. How many coils? 1.5 coils. You coil it around histone proteins. Histone is, how many protein molecules are present in the histone core there? 8. It is a histone octamer. It is a histone octamer. 8 molecules of histone are present. When you coil it, when you coil the DNA around the histone, now that unit of DNA coiled around histone is called nucleosome. The entire length of DNA with coiling of the DNA around the nucleosome at periodic intervals. This entire length of DNA is called chromatin. Chromatin. You fold the chromatin and condense it. Then you call it, what do you call condensed chromatin? Chromosome. Now this DNA replication happens. That means the DNA becomes double. It becomes 2 strands. Both the strands remain together and it condenses. What is this? These 2 are exactly the same strands. What is this? Huh? What is this? These are the 2 strands of DNA. They condense and remain together. What do I have now? This entire thing? Chromosome. What does each one of the index finger represents in this chromosome? Chromatid. What kind of chromatid? Sister chromatids. Now I separate these 2. What do I have? What do I have? 2 chromosomes. How many chromatids do I have? How many chromatids do I have in these 2 chromosomes? 0. Chromatids are only those which are condensed chromatin, which are a part of the same chromosome. Which are a part of a chromosome. When the 2 condensed chromatin are part of the chromosome, you call it chromatids. But when you separate it, it is still chromosome only. So the definition of chromosome is a unit of condensed chromatin. Polytene chromosome. What is polytene chromosome? Polytene chromosome. Polytene. Polytene chromosome. Found in the salivary glands of which one? Drosophila? What is polytene chromosome? Polytene chromosome is a giant chromosome. Remember polytene chromosome. Polytene, giant chromosome. It will look like our Ashoka Chakra. Where do you find Ashoka Chakra? In Indian flag. How many spokes are there? 24 spokes. So you consider the radial spokes or the diameter? Radial spokes, 24 radial spokes. Polytene chromosome looks like that. Multiple chromatids are present. More than 2 chromatids is giant chromosome. Polysome or giant chromosome. Polysome is multiple chromatids in a chromosome. More than 2. Got it? Okay, now you answer this question. Association of histone with a nucleosome indicates what? DNA replication is occurring? No. DNA is condensed into a chromatin fiber. That's the right choice. More than 80% of the questions in biology can be answered by common sense logic. More than 80% of the questions. Alright? Trust me and stick to the papers I discuss and the last 12-13 papers. Master that much, you will cross 300. Biology alone. Alright, 80% of 360 is how much? 288. 288. The first 13 papers, if you master all the previous year papers, if you master the logic of the first 13 papers, 288 you will cross. The last 13 papers which you will attempt, master that much, 300 to 320, you will certainly cross, only in biology. Okay. Same you do with physics and chemistry also. Another 200 to 250 with physics and chemistry put together, you will cross. 500 to 600, easy. Okay. I don't tell this in the beginning because I want you to go through all the things that you have gone through. That will save you forever. The process that you have been trained will save you forever. Exam is only this much. Master 10 papers. 10 papers. By the right approach, if you master 10 papers, you will clear the exam. Don't get stressed out. Don't get overwhelmed. Alright. I have been training students in, cracking these exams for a duration that is more than your age. The same nonsensical statement that every old man gives to younger people. My experience is more than your age. Okay. But it means something, so don't get overwhelmed. The biggest challenge for you in these 76 days is to stay grounded and focus on your preparation. Don't get overwhelmed by fear, anxiety. Alright, don't get overwhelmed by the volume of content that you have to master. Clearing exams and being knowledgeable are not necessarily the same things. Alright. You need not be very knowledgeable to clear exams. But you have to be knowledgeable to get respect in your professions. You have done both. You have worked on the process that will make you knowledgeable. You have used that process to build on the fundamentals for your preparation. Now, in the final phase of this preparation, I am telling you what will help you clear the exams. Stick to it. Alright? So, the more happy you are, the more excited you are to go and face the exam, to test yourself. Come on. It's like, right? Have you watched any Charlie Chaplin movies? In one of those movies, he is put in the ring with a boxer or wrestler. He is not a fighter. He has never trained, or his muscles are like that and he is like that. But what does he do? Survival instinct kicks in. He wants to survive. That's all. He does not want to die because he knows that if he does not tackle this fellow, he will die. So he takes a commonsensical approach. What is the commonsensical approach? I cannot beat him. I'll escape his beating. I'll not get into his hands. So he escapes, he ducks. And by the end of it, that fellow trying to catch Charlie Chaplin, he gets so tired, That one blow of Charlie Chaplin makes him further, and he is like, better I get out of this ring. And he wins, no? Charlie Chaplin wins the game, wins the bout. That's all. Okay, so activate your common sense, activate your logic. Don't try to recall, have I learned this, do I remember this? Activate your common sense. Alright, the more you keep responding, come up with some logic to answer the questions. All these are papers which you already attempted, but try and come up with some logic to give a justification to your choice during these practice sessions. You will do well. So something should come out of your mouth, not the yarn. I'm telling something should come out of your mouth, and this fellow okay. Right, come on, next one. These kind of questions I can't help you. Which one from those given below is the period for Mendel's hybridization experiment? I don't know the period of my grandfather. 1856 to 1863. Skip. Root hairs develop from the region of maturation. The regions cap, elongation, or cap, meristematic activity, elongation, maturation. All the functions are found in the maturation region. In Bougainvillea, thorns are the modifications of stem modification. Stipule is a modification of what? Stipules. Is it a leaf modification or a stem modification? Stipules are also coming from the base of the leaves, like thorns. Right. Stipules also can give you protection. They can do photosynthesis, all of that. They are also coming from the same node from which the leaves are coming. Stipules can be photosynthetic. So, what modification do we consider that? We can still consider that also to be the stem modification only, no? Stipules. They are leaf-like structures. You can call them modified leaves also. DNA replication in bacteria occurs within nucleolus. There is no nucleolus. Just before transcription. Transcription is not associated with DNA replication. Transcription is associated with mRNA production. During S phase, S phase is a part of DNA replication, is a part of the process of DNA replication. So S phase is also correct. DNA replication in bacteria occurs prior to fission. That is also correct, no? Prior to fission, before the cell divides, DNA replication occurs. Which is the phase of mitosis when DNA replication occurs? That would be S phase specifically. But DNA replication, we should not go into the phase of mitosis where DNA is replicating, when the phase in the life cycle of the cell which talks about DNA replication is present as an option. S phase is also technically correct, but the appropriate choice here would be before fission, prior to fission. Water potential of pure water is, now again coming back to this, potential and pressure. Which of these two encourages osmosis? Potential or pressure? Which of these two encourages osmosis? Which of these two will give the power to a cell to give water? Potential helps you give water. Pressure forces you to take water. Pressure is a force. Pressure forces you to take water. Potential gives you the ability to give water. Higher potential increases exosmosis or endosmosis? Higher potential increases exosmosis or endosmosis? Exosmosis, because it gives. Higher the potential, more water you give. Higher pressure increases endosmosis. Higher pressure increases endosmosis. Turgidity of the cell increases when there is higher water potential or higher osmotic pressure. Higher osmotic pressure will lead to increase in turgidity. It leads to increase in turgidity. So remember all of this. Potential pressure, understand it. Highest potential of any system containing water is for which kind of water? Pure water is, what is pure water? Is this pure water? This is not pure water. Pure water is what? The first 2 days when I come here, my body goes into a detoxification mode. And suddenly my muscles will start cramping. So many things happen. My body is not used to inhaling oxygen at higher partial pressures. It is used to taking oxygen with a lot of pollutants. Partial pressure, no? Oxygen absorption happens under partial pressure gradient. Here partial pressure gradient is very high for oxygen. Less pollutants. My body doesn't like it. We are not used to that. We are used to breathing all sorts of nonsense with here. So now my back muscles have cramped. Not so much cramp now, otherwise I would be walking like a duck. Okay. I'll tell you some potentials. You tell me, does it promote exosmosis or promote endosmosis when it is high? Or you have to tell me, okay, water potential, highest water potential that a system can achieve is 0. Highest water potential that a system can achieve is 0. Water potential in any system is 0 or negative. There is no positive water potential. You understand that? Unmanipulated systems. Unmanipulated systems. If you exert pressure, then you can create a positive water potential. That is a different issue. If I add, okay, what is pure water? This is not pure water. Then what is pure water? Chemically, when we talk about pure water, what is it? Water with 0 solutes. Water with 0 solutes is pure water. How do we make this pure water? How do you convert this into pure water? Reverse osmosis, okay. I will not give you any such systems. You have to convert this into pure water. Boil this water. When you boil water, what happens? Steam. Allow the steam to condense. What do you call that water? Distilled water. Distilled water. Distilled water is pure water, 0 solutes. So the water potential in that is 0. Even if you add a little bit of solute, the water potential will become negative. Because solutes will tend to pull water, no? The highest potential for pure water is when it is absolutely given. Solute potential increases endosmosis or exosmosis? Endosmosis. It is also referred to as osmotic potential. Osmotic potential or solute potential. Pressure potential. Pressure potential. See, a cell, a plant cell, let us say, with more and more water being taken in. Let us say there is a lot of solute inside the cell. It will start taking in a lot of water. It will start taking in a lot of water. The turgidity of the cell wall will prevent more water from coming in. That is called turgor pressure. It will prevent more water from coming in. So is turgor pressure complementary to osmotic pressure or is it opposite of osmotic pressure? Opposite, because osmotic pressure is trying to draw water into the cell. Turgor pressure is preventing water from coming into the cell by preventing the expansion of this cell. Alright. So how much water can come in or how much water can the system accumulate is equal to osmotic pressure plus turgor pressure. Both of them put together. Turgor pressure is positive. Osmotic pressure is negative. Okay. Anyways, remember this. Water potential, solute potential, osmotic potential, osmotic pressure. Osmotic potential and osmotic pressure are opposite. Osmotic potential is the ability to give. Osmotic potential and water potential are the same. Okay, there is one more concept. Diffusion pressure deficit. Diffusion pressure deficit. Water moves from one point to another point. Let us say the pressure with which water moves is the diffusion pressure. Diffusion pressure. So higher the diffusion pressure, higher is the ability of the water to move out of that system. Now can I say diffusion pressure is equal to water potential? Can I say diffusion pressure is equal to water potential? Both are the ability. Water potential is talking about the ability to give. Diffusion pressure is talking about the the force which is driving water out of the system. Both are same. You are talking about it from a different perspective. Understanding, not understanding? Who is not understanding? Who is understanding? I experience this kind of humiliation when I address parents or older people. Generally, younger audience is more responsive. I address a lot of parents. I tell them I know how to support your children in education and all that. The opening approach of the parents is, who are you to tell? Don't we know our children? Do we know our children better, or you know our children better? So they'll be sitting like this. That's our entire session. They will be sitting like this, many of them. Some of them I will be able to open them up, but yeah. Pens are also mimicking your response. Alright. The last one. Oh. Now these pens are behaving like the switches on the unknown room. Whenever you go into an unknown room, there are 4 switches. You want to turn on the light. Mostly the last switch is the one which is the right switch. Sometimes you go from this to this side, it is the last switch. Next time you have learnt your lesson, you go from the other side, again that is the last switch. Okay. Psi is the symbol. This is the water potential. Water potential will help in movement of the water from inside to outside. So higher the water potential, higher is the tendency. Solute potential. Solute potential is negative. Solute potential, again it's also, whenever we talk about potential, it is moving out only. Moving out only. Then you have pressure potential. Pressure potential. So in any system, water potential is a combination of solute potential plus pressure potential. Pressure is how much is the pressure that is exerted on the system for movement of water. This can be, water potential can be 0 or negative. Solute potential can be only negative. Pressure potential can be positive. Pressure potential is positive. Alright? You don't have a negative pressure potential. Pressure is exerted on a system which will increase its potential to give water. So in a system which is, this can be zero also, where you have no pressure exerted on the system. So in a system where you are not applying any pressure externally, in a system where you are not applying any pressure externally, water potential is mostly equal to the solute potential. Water potential is mostly equal to the solute potential. Water potential is zero. Water potential is zero in pure water. Water potential is zero in pure water. Now if we have two systems, between which water moves, let us say water moves from system A to system B. Water moves from system A to system B. This has a higher water potential and this has a lower water potential. Water moves from a system of higher water potential to a system of lower water potential. The one who has higher potential to give is the one who gives, and the one with lower potential to give is the one who receives. That's all. Common sense. The one which has a higher diffusion pressure will give to the one which has a lower diffusion pressure. Diffusion pressure. That concept I want you to understand. It is diffusion pressure. How much it moves is dependent on the diffusion pressure deficit. This minus this is the diffusion pressure deficit. It can also be water potential gradient or water potential difference. Diffusion pressure deficit can also be the difference in the water potential, no? The one with the higher diffusion pressure, let's say 10 units of diffusion pressure. The other one has 2 units of diffusion pressure. So the diffusion pressure deficit is 8 units. Another system has, let's say, 6 units of diffusion pressure. The fourth one has 4 units of diffusion pressure. Between A and B, you have 8 units of diffusion pressure deficit. Between C and D, you have 2 units of diffusion pressure deficit. Which one will have a higher movement of water? A to B or C to D? A to B. More the deficit, more the gradient, more is the movement. Yes or no? More the gradient, more is the movement. Lesser the gradient, lesser is the movement. Diffusion pressure deficit will tell you the intensity with which movement or diffusion of water happens from one system to another system. Questions might come on comparative systems. You will be given 2 systems with diffusion pressure gradient and water potential difference, one with more solute potential, one with less solute potential. All of those will be given. Irrespective of whatever is given, the question will be related to the movement of water from which one to which one, or which one will have a higher movement or lower movement. That's all. Question will be related to movement only. There are multiple potentials. Remember a few: water potential, solute potential, pressure potential, diffusion pressure deficit. If you know the logic behind all this, whenever the question is asked, you can think, you can you can jot down some things and come to a conclusion. Okay. Don't try to memorize these things. Remember this. Primary stain is methylene blue. Primary stain is methylene blue used for staining the cell wall. If the bacteria is taking up the primary stain, what does that imply? Cell wall is being stained. Cell wall is being stained. That means no capsule. If primary stain is not taken up and you add secondary stain, safranin, and the secondary stain is taken up by glycocalyx. Peptidoglycan takes up methylene blue. Glycocalyx takes up safranin. So if methylene blue is not taken up, what does that mean? The stain is not coming in contact with the cell wall. That implies there is a capsule, no cell wall. Cell wall, it cannot reach the cell wall. Therefore, you add the secondary stain. Then it takes up. What does that mean? Capsule is present. If there is a capsule, capsulated bacteria are gram positive or gram negative. capsulated bacteria are gram positive or gram negative? See, repeat questions, students. All these are all repeat questions. It might come in this form or that form, that's all. But the questions, how many concepts do you have? You have only limited number of concepts to ask questions from. Just to make it different, you cannot, the people who set papers cannot give you some random things. They have to give it from the same thing. Only thing is it will be twisted. Oh, the zinc here. Gram positive. This will not move so easily. We should leave this to rest. There are no other pens, madam? Get some pens. Okay, this is the cell wall. Consider this as the capsule. Okay, the outer one is the capsule. The outer one is the capsule. The inner one is the cell wall. When you add the primary stain, if there is a capsule, it cannot cross the capsule and stain the cell wall. It cannot cross the capsule and stain the cell wall. And this primary stain will not stain the capsule. So primary stain will not be taken up. If primary stain is not taken up, then what do? You wash it again and add the secondary stain. Secondary stain will bind to the capsule. Secondary stain will bind to the capsule. So if secondary stain is taken up, then it is gram negative. When we say gram positive and negative, we are talking about whether the bacteria can take up primary stain or not. If it can take up the primary stain, we call it gram positive. If it doesn't take up the primary stain, we call it gram negative. Why is it not taking up the second primary stain? Because it has a capsule. Because it has a capsule, it cannot take up the secondary stain. Alright. It is like finding out whether you are inside the room or outside the room. Let's say there is a transparent shell. There is a transparent shell. How do I find out whether you are inside a transparent shell or outside a transparent shell? I will throw water on you. Okay? If the water wets you, then you are not having the shell around you. If the water does not wet you, then you have a shell around you. How do I know whether there is a shell again? Water is not wetting you. Second time I will throw colored water at you. It will color the shell. That is the secondary stain. Understand? So I call you positive or negative based on whether you are whether water can reach you or not. If the what is the shell now here? The shell is capsule. Shell is the capsule. That's all. Gram negative bacteria are more pathogenic. They can cause more severe diseases than gram positive bacteria. Understand? Final proof for DNA as the genetic material came from the experiments of, whose experiments gave the final proof of DNA as the genetic material? Hershey-Chase experiment. Griffith is known for what experiment? Transformation experiment. Avery, McLeod and McCarthy, DNA. DNA as the genetic material. The DNA fragment separated on an agarose gel can be visualized after staining with. What do you use for staining DNA in the agarose gel electrophoresis? Ethidium bromide. Is ethidium bromide. What is the charge on DNA? Negative charge. Negative charge is because of what? Phosphate group. Phosphate group gives the negative charge. So it moves towards what on electrophoresis? DNA moves towards anode. Anode. So on which side of the towards the well, what charge do you apply? Towards the well, what charge do you apply? Negative charge. So on the opposite side you have the positive charge. So DNA moves from the negative to the positive side. Alright. What is the criteria for DNA fragment movement on agarose gel during electrophoresis? The same one again. Smaller the fragment size, farther it moves. Commonsensical. The other four are obviously wrong choices. Positively charged fragments move to the farther end. DNA all fragments are negatively charged. Negatively charged fragments do not move. Larger the fragment size, farther it moves. No. Which of the following options give the correct sequence of events during mitosis? Look at all the four. Condensation is the beginning, telophase is the end for all the four. Correct? Condensation is the beginning and telophase is the end. Okay. Nuclear membrane during mitosis. We need to fill ink to these things. Condensation is the beginning, telophase is the end. Nuclear membrane disassembly, arrangement at equator, centromere division. See, anything that mentions crossing over, rule out. Why? Crossing over is not a part of mitosis. So option 2 and option 4 ruled out. Between option 1 and 3, look at the difference. Condensation, telophase, nuclear membrane disassembly, arrangement at equator, centromere division, separation of chromatids. Option 3 talks about segregation before arrangement at equator. Segregation happens in which phase? Anaphase. Arrangement at equator happens in metaphase. So 3 is wrong, 1 is right. Again, nothing to think too much, just organize it, that's all. Which of the following are found in extreme saline conditions? Archaebacteria. There are 3 types of archaebacteria we study. What are the 3 types? Halophiles, thermoacidophiles and methanogens. Eubacteria, cyanobacteria, mycobacteria, all of those are bacteria. All of those are eubacteria only. Cyanobacteria is algae. Which among the following are the smallest living cells known to lack a definitive cell wall, are pathogenic to plants as well as animals and can survive without oxygen? Which one it is? Mycoplasma. Why not Pseudomonas, Bacillus, Nostoc? All of them have cell wall. Pseudomonas and Bacillus are bacteria. They have cell walls. Nostoc is a cyanobacterium. That also has cell wall. Mycoplasma is pathogenic and no cell wall. Fruit and leaf drop at early stages can be prevented by the application of auxin. Auxins help in preventing fruit drop or leaf abscission. Drop is abscission. Ethylene promotes abscission. Which of the following is currently correctly matched with the product produced by them? Methanobacterium lactic acid. The name itself says it produces methane. Penicillium notatum produces penicillin. Acetic acid is produced by Acetobacter aceti. So 2 and 4 are interchanged. The right choice is Saccharomyces cerevisiae, which produces ethanol. See, again in this chapter, examples, that's all. Nothing more than that. Processes are all very simple. Which of the following facilitates opening of stomatal aperture? Decrease in turgidity of guard cells. Is it right? Guard cells open when the turgidity of guard cells increase. Stomata opens when the turgidity increases. Radial orientation of cellulose microfibrils in the cell wall of guard cells. Longitudinal orientation of cellulose microfibrils in the cell wall of guard cell. So 2 and 3 talks about microfibrils contributing to guard cell opening. One is talking about the radial arrangement and the other one is talking about longitudinal arrangement. How are the microfibrils arranged in guard cells? They are arranged radially. Therefore, the contraction turgidity pulls them away from the stomata. Flaccidity makes them get closer to the stomata. Makes the guard cell come closer to each other. So radial arrangement, radial orientation is the right choice. Which statement is wrong for Krebs cycle? There is one point in the cycle where FAD is reduced to FADH2. Is it wrong? Is it wrong? It is correct. Can you tell me the step where FAD is reduced to FADH2? Succinate to fumarate. Succinate to fumarate. Can you tell me the sequence of reactants in Krebs cycle? Starting with oxaloacetate combines with acetyl-CoA to form citrate. After that, isocitrate. Then alpha-ketoglutarate. Then succinyl-CoA, succinate, fumarate, malate, oxaloacetate. SFM. Number of carbons. Acetyl-CoA, 2 carbon. Oxaloacetic acid, 4 carbon. Citrate, 6. Isocitrate, 6. Isocitrate is 6 again. Alpha ketoglutarate, 5. Isocitrate to alpha ketoglutarate, you are losing 1 carbon dioxide. After alpha ketoglutarate to succinyl-CoA, 4. Then rest all are 4. During conversion of succinyl-CoA to succinic acid, a molecule of GTP is synthesized. True or false? True. The cycle starts with condensation of acetyl group with pyruvic acid to yield citric acid. False. What is the right thing there? Condensation of acetyl group with oxaloacetic acid, not pyruvic acid. There are 3 points in the cycle where NADPLase is reduced to NADH. Correct. Okay. A gene whose expression helps to identify transformed cells is known as selectable marker. A dioecious flowering plant prevents both. See, first of all, dioecious means what? Unisexual plant or a plant with unisexual flowers? Unisexual plant. Dioecious is a reference to unisexual plant, not unisexual flowers. Okay. So, what is not possible in a unisexual plant? Self-pollination, autogamy is not possible. Autogamy has 2 types, cleistogamy and geitonogamy. Cleistogamy is closed flower. So the same flower pollen fertilizes the flower. Geitonogamy is pollen from a different flower of the same plant pollinates it. So both are not happening in in a dioecious plant. Now look at the options: autogamy and geitonogamy. Geitonogamy and xenogamy. Xenogamy is cross pollination. So if xenogamy is there, then that is what happens. in a dioecious plant. That's all. So it's very obvious. The right choice is autogamy and geitonogamy. Flowers which have a single ovule in the ovary and are packed into an inflorescence are usually pollinated by bee, wind, bat, water. Wind pollination. See, flowers packed into inflorescences in inflorescence is increasing the surface area for pollination. Surface area for pollination. Correct? So why do you have to increase surface area for pollination when the pollinator is non-specific? When the pollinator is non-specific, you have to increase the surface area to increase the chances for pollination, to increase probability. Correct? No? When the pollinator is specific, what do you have to do? You should create mechanisms for the pollinator to be attracted towards the flower. So specific pollinators are insects, bee, bat, these are all specific pollinators. You have to give them some incentive to do what you want them to do. Wind is non-specific. It will just carry the pollen. So when the wind is carrying the pollen, the chances of pollination increases when the surface available to capture that pollen is high. So it does you good when you have an inflorescence loading up the flowers instead of individual flowers. So wind pollination, again common sense. Approach it with common sense, you will get answers. Ask questions, why, what, when, where, these kind of questions when you ask, you will get answers generally. Attractants and rewards are required for, again see here, again same question, continuity. Attractants and rewards are required for entomophily, hydrophily, cleistogamy, anemophily. Entomophily is insect pollination, hydrophily is water pollination, cleistogamy is closed flower, you don't need mediators for that. Then anemophily is wind. Anemophily is wind pollination. What happened? Animals, anemophily is not animals, anemophily is wind. Alright. See, it's like, which one is more successful of the four? Entomophily, hydrophily, cleistogamy, anemophily. Which one is more successful? Cleistogamy, no? No, more prevalent. What do you find? Cleistogamy eventually will lead to decrease in the genetic viability and the vigor. Entomophily is the most beneficial, no? Correlate all of this before we take a 10-minute break. Correlate all of this to the business scenario. Eventually, you will all get into the economy. You will do business. Even if you are in a job, it is a business setting. Cleistogamy is like a one-person restaurant. He is the one who cooks. He is the one who serves. He is the one who collects cash also. Is it scalable? If that person is ill, the restaurant is shut for the day. Doesn't trust anybody. Cleistogamy is a situation where you don't trust anybody. You feel you are the only one who can do the right things. You might be more efficient, but if the entire business is dependent on only you, it is not sustainable. It will die with you. Anemophily is there is a big, let's say a mela happening, public place. Let's say you go to a beach, you go to a tourist spot. Are the people who are selling there trying to establish loyalty with you? Do they want you to be loyal? No. The approach is get as many people as possible. They cheat you, they overcharge you, they look at you and decide how much price to tell. Correct? Anemophily. Increase the chance by exposure to a large number of people. Wind. No loyalty required. reward and all the incentives, nothing required. Just because even if you might curse them, your business will get destroyed. At last, you will not come tomorrow. Tomorrow somebody else will come and they will also tell the same thing. It doesn't matter to me. Anemophily. Increase the chances of business by increasing your surface area. Have you gone in trains? On the platform there will be a small store. He will be selling idli, vada, or samosa, whatever, depends on the place where your train is stopping. There will be people inside the train coming with trays of the same food. They will be carrying the same food in the tray. Samosa, 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 garam, garam, samosa. Garam, garam, chai, 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 chai. It is the same store there. Why is he employing 10 more people to go into the compartments, to go into the bogies? Increase exposure, anemophily. The more you expose the potential customers with the product, the more are the chances of transaction, which will increase your sales, which increases your profitability. Depends on what kind of business you are setting up. Entomophily is like a proper business, established business. You need to create a loyal customer base. Give rewards, give some reasons for people to buy, build loyalty, all of that. Otherwise, insects will not come. Alright, it's like that. Anyways, attractants and rewards are required for entomophily. Entomophily wants to, when it is entomophily, you have to put out banners. The best outcomes comes with us. Best results. See, the topper comes from our institute. Entomophily, attractants, rewards. Alright, so higher the reward, more price the insects will pay. Correct? You have seen this Venus flytrap? Venus flytrap? Very attractive, very juicy nectar. More rewards, more rewards. The insect is made to believe that its life will change if it gets a taste of this. The taste, just the taste of it will change your life. So what the insect will do? Insect will try to go deep into that. Just an inquiry is enough. You just download the app. The next day, if somebody will be in your house. Have you experienced this? You download one app. Tomorrow you get a call. You have downloaded our app. You have gone through this thing. This is your analysis. If you are ready, I will give you a full demo of the product now, right now. Or if you want, I'll come to your house. When? In 10 minutes. I can come faster than the ambulance. Venus flytrap. Nature tells you how to do business. Alright, anyways. Okay. Entomophily. We'll take a quick 5 minutes break. Come back. Now it is 11:05. No sunbathing for anybody alone. If I find people doing sunbathing there, what will I do? I'll tell somebody to call you. How many megaspores degenerate? 3. The one which does not degenerate is referred to as functional megaspore. It eventually transforms into embryo sac. By how many cycles of mitosis? 3 cycles of mitosis. 3 cycles of mitosis. Okay. Embryo sac has how many cells? 7 cells. What is the ploidy of the embryo sac as a whole? Assuming the parent was diploid. Ploidy of the embryo sac as a whole, assuming the parent plant was diploid. Haploid, ploidy of the embryo sac. Ploidy of the embryo sac. How many sets of chromosomes are present in the embryo sac? If we assume embryo sac to be a single cellular entity, What is its ploidy? Huh? Octaploid. 8 sets of nuclei are present. How many cells are there? 7 cells. 6 haploid and the central cell is diploid. Central cell is diploid. Should we call the central cell as diploid or a dikaryon? It should be a dikaryon, no, because it has 2 nuclei. It has got 2 haploid nucleus. A diploid cell is the one which has a single nucleus with 2 sets of chromosomes. Okay. So whatever can be the question, twist and turn, basic is this. Double fertilization is exhibited by algae, fungi, angiosperms, gymnosperms. Double fertilization is a characteristic of angiosperms. Double fertilization is also associated with another process, triple fusion. Triple fusion happens where? In which cell is the one which is participating in triple fusion? Central cell. Correct? No, double fertilization happens with the ovum and the central cell being fertilized. But triple fusion is associated with the central cell. Triple fusion gives rise to what? Triple fusion gives rise to endosperm. Triple fusion gives rise to endosperm. Alright? Which of the following is an outcome of double fertilization and triple fusion? Can be the question. Options can be embryo, cotyledons, endosperm, perisperm. Which one do you choose? Endosperm, no? Triple fusion is happening in the central cell and the central cell eventually develops into an endosperm. Cotyledon is also developing from the fertilized egg which becomes the zygote, which gives rise to both embryo as well as the cotyledons. Okay. What is pericarp? Pericarp. 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 Carp is what? Carp, C-A-R-P. Carp is a reference to carpel. Carpel is a reference to what? Ovary. Ovary eventually develops into what? Fruit. So carp, can it be a reference to fruit? Peri means what? Covering or boundary. So can pericarp be the fruit wall? Pericarp can be the fruit wall. Correct? Pericarp is the fruit wall. There are 3 layers in that. There are 3 layers. Mango is a very good example of that. Mango, raw mango, not the ripe one. If you cut raw mango longitudinally, you will find the outer skin, the middle light green part which is the raw mango which you use for pickle and all. And there is that inner shiny fibrous part. On the inside of the fruit, there is that shiny fibrous part, almost translucent. That is also a part of the pericarp. There are 3 layers now. Outer dark green part, the skin. The middle fleshy or a bit hard and fleshy mango. And the innermost shiny harder part. And then you have the seed. Correct? Many times when I eat that mango pickle, I feel that why don't they remove this, the inner one? When you eat it, it becomes hard. You cannot eat it. It is very fibrous. So those are the 3 layers. The outer green one, the middle fleshy one, and the inner shiny one. All 3 together is pericarp. Only the outer green one is epicarp. The middle part is mesocarp. The inner shiny one is endocarp. Then you have the seed. You have the seed which you remove and throw it off. Seed coats are different. The inner shiny one is not the seed coat. It is the inner layer of the fruit wall. Mango is the best example there. So pericarp has 3 layers. What are the 3 layers? Epicarp, mesocarp, endocarp. Similarly, there is one more which we refer to as what? Perisperm is what? Perisperm is a persistent nucellus. Perisperm and endosperm are common with respect to their function. What is the function for both of them? Provide nourishment to the developing embryo. Both are nutritive tissue, perisperm and endosperm. But perisperm is diploid, endosperm is triploid. Alright? One more peri. Somewhere else we find one more peri. What is periderm? Periderm. Periderm is bark or cork. If you consider it as a set, which one is the bigger set? Which one is a subset? Bark or cork? Which of these two, between bark and cork, one is a subset of the other. Which one is the subset? Bark or cork? Huh? Cork, huh? Cork is subset. No? Bark is the subset. Okay, which one is the subset? Bark is the bigger one. Bark is made up of cork, cambium, and secondary cortex. All 3 together is bark. So bark is periderm, phellem, phellogen, phelloderm. You get that? Very important. Questions can be confusing there. Okay, we'll get that question and we'll do that. At which of the following stages of sewage treatment are suspended solids removed? Do you want to remove suspended solids from sewage at the initial stages of treatment or later stages of treatment? Initial stages. So the initial treatment is primary treatment. Okay. Phosphoenol pyruvate is the primary carbon dioxide acceptor in C2 plants, C4 plants, C3 and C3 plants. C4 plants. Who is the primary carbon dioxide acceptor in C3 plants? Rubisco. Rubisco accepts it, then it becomes phosphoenol pyruvate or 3-phosphoglyceric acid. Phosphoglyceric acid is what is formed. Phosphoglyceric acid goes into the Calvin cycle for photosynthesis. But in C4 plants, who receives the carbon dioxide? Who receives the carbon dioxide? PEP carboxylase. Phosphoenol pyruvate carboxylase will combine carbon dioxide with phosphoenol pyruvate and convert it into oxaloacetic acid. Oxaloacetic acid is converted into malic acid. Malic acid will be transported to bundle sheath cells, stored there, and during the daytime malic acid is transported into the mesophyll cells where it is broken down. Reverse of this happens. Whatever had happened, reverse of this happens. Malic acid to oxaloacetic acid to pyruvate and carbon dioxide. Carbon dioxide goes into the Calvin cycle begins there. Calvin cycle happens there. Alright. So 2 types of cells are associated with photosynthesis in C4 plants. What are the 2 cells? Bundle sheath cells and mesophyll cells. In C3 plants, it is only mesophyll cells. Mesophyll cells surrounded by bundle sheath cells is a special structure. What anatomy is that? Kranz anatomy. Kranz anatomy. So remember Kranz anatomy with C4 plants. Kranz anatomy is seen in plants which are predominantly found in tropical zones, temperate zones, equatorial zones. Tropical zones. C4 C4 pathway is found in tropical plants. C4 pathway is an adaptation to prevent the plant from dying due to excessive transpiration during the daytime when there is too much of heat. But without opening the stomata, carbon dioxide cannot get inside. Correct? Without carbon dioxide coming inside, no photosynthesis. If you open stomata, water loss. To take carbon dioxide to manufacture food, if you open stomata during the daytime, excessive water loss and the plant will die of water loss. If you don't open stomata, no carbon dioxide enters the mesophyll cells, photosynthesis cannot happen, you have to die without food. Now, nature has given a choice to the plants in the tropical areas to die without food or to die without water. What did the plants do? They created a third option, neither. Alright, redesign and develop adaptation to survive. Develop adaptations to survive. So sometimes you will be faced with two choices, both of them damaging. At that time you have to have courage to chart out your own third path, discarding both. Alright? So, do you want to do engineering or medicine? No, now you are clear with that because you have gone too far in this path. Somebody is very artistic, somebody is a good singer. The child will be given two options after 10th standard. Do you want to take up engineering or medicine? Now the child thinks, oh, these are the only two options available. They will think about which option will give me opportunity to pursue my singing, and then they will choose that. Some brave people will say, neither. I will take up arts, I will go into performing arts school, I will pursue music. Suddenly the world falls on the head of parents. Oh, music as a career. Oh my God. Nothing happens. At least now people are more open to thinking because so many career options where people are earning loads of money which parents don't even understand can be career options. What career? What is this? For a very long time I did not understand what software engineers do. I used to ask my friends, so what will you do? I will write code. So, now you understand, everything is code. You walk in, suddenly light is turned on, and you walk out, light turns off. You put your hand in front of the tap, water starts coming. Take it out, water goes off. All magic happens. Okay, C3, C4. So C4 plants, Cran's anatomy, bundle sheath cells, mesophyll cells, pep carboxylase, that's all. Photosynthesis is split, day and night photosynthesis happens. So light independent reactions, part of it happens in the night and completion happens in the daytime. But in C3 plants, even light independent reaction happens during the daytime only. Which of these two consumes more ATP? C3 or C4? C3 or C4? C4. 18 in C3, 30 in C4. Select the mismatch. Cycas, dioecious, Salvinia, heterosporous, equisetum, homosporous, Pinus, dioecious. Pine has both male and female cones. Pinus coniferous, no? Pinus is gymnosperm. Has both male and female cones on the same plant. So Pinus is monoecious, not dioecious. Coconut fruit is a berry, nut, capsule, drupe. This chapter is only not there, no? This is morphology. What is a drupe? Coconut is a drupe. Fleshy, outer fleshy part and a hard seed inside. Coconut and mango. What are the three layers of the fruit layer? Exocarp, mesocarp, endocarp, All three put together is pericarp. Okay. Vascular cambium normally gives rise to primary phloem, secondary xylem, periderm, phelloderm. Now see this cambium, vascular cambium gives rise to secondary growth. Vascular cambium gives rise to secondary growth. It is secondary meristem. Alright, cambium is meristem. So primary phloem is not the one which comes from secondary growth. Primary phloem is primary growth. Secondary xylem is correct. Periderm and phelloderm are not associated with cambium activity. Periderm also has a secondary vascular tissue. What is the secondary vascular tissue? Secondary cambium that you find in periderm? Phellogen, which is cork cambium. That is also part of the secondary meristem. Phelloderm is secondary cortex. So secondary xylem is the right choice. Different types of cambium. Vascular cambium. Vascular cambium is present between xylem and phloem. Gives rise to secondary xylem and secondary phloem. Then you have cork cambium, phellogen, gives rise to secondary cortex. Cork cambium gives rise to secondary cortex. Then based on the position of the cambium, you have intrafascicular cambium. Intrafascicular cambium is within the bundle, within the vascular bundle, between xylem and phloem. Then between the vascular bundles, what do you call it? Interfascicular cambium. Fascicle means bundle. Alright. Which of the following is made up of dead cells? Easy question. Collenchyma, phloem, xylem parenchyma. Xylem parenchyma is the only living component of xylem. Collenchyma is living tissue, living simple permanent tissue. Phloem is cork. Cork is dead tissue. Phloem. Does phloem not contain dead cells at all? Phloem also contains dead cells, which are phloem fibers. There are 4 components of phloem. What are the 4 components of phloem? What are the 4 components of phloem? Sieve tubes, companion cells, phloem parenchyma and phloem fibers. Sieve elements you can say, sieve tubes and sieve cells both. Sieve elements, companion cells, phloem parenchyma and phloem fibers. Phloem fiber is dead, all the other 3 are living. Xylem also has 4 components. What are the 4 components? What are the 4 components? Tracheates, vessels, that is trachea, then xylem parenchyma, xylem fibers. Of these 4, only the xylem parenchyma is living, all the other 3 are dead. So xylem has 3 dead and 1 living component. Phloem has 3 living and 1 dead component. Xylem transportation within xylem is mechanical. Transportation within phloem is cellular, controlled. Okay. Identify the wrong statement in the context of heartwood. It is highly durable. True or false? Highly durable. Heartwood is the one which is used for all the furnitures. Furniture, for construction, all that is heartwood. So heartwood is the one which is condensed. Lot of resin and other inclusion materials within that. Durability is high. Is it living? Will you have living components or functional components in heartwood? There are no functional components. What part of the wood is having functional components? Sapwood. Heartwood is also referred to as hardwood. It is hard. Sapwood is also referred to as softwood. Both of these are a result of secondary growth. Now, which one is wrong? It is highly durable. It is correct. Conducts water and minerals efficiently. That is wrong. There is no conduction at all in heartwood. Comprises dead elements with highly lignified walls. Yes. Organic compounds are deposited in it. True. Select the mismatch. Rhodospirillum Rhodospirillum is sulfur bacteria. It is not associated with mycorrhiza. What are mycorrhiza? Association of fungus with roots. Fungus helps in absorption of nutrients, minerals, breaks down minerals and makes it available for the plant roots. Roots provide shelter to the fungus. Anamina nitrogen fixer, it is correct. Rhizobium is also a nitrogen fixer and it is found in alfalfa. Frankia is found in alnus. It is a symbiotic nitrogen fixer again. Symbiotic nitrogen fixation. Process of separation and purification of expressed protein formed in the heterologous host before marketing is called downstreaming. Do you notice in the last 5 papers including this is the second or third time that a question is coming related to downstreaming. What else you have? Biotechnology, the application of biotechnology. You have this only, no? Zygotic meiosis is characteristic of zygotic meiosis, meaning what? Meiosis happening in zygote. What does that mean? Wherever, in whichever organism you find zygote undergoing meiosis, what can you infer from that? What inferences you can make? Meiosis is happening, therefore the organism is having a sexual reproduction as a part of its reproduction. Okay, then if zygote is undergoing meiosis, what does that mean? Gametes are produced by mitosis. Gametes are produced by mitosis. If there is a need for gametes to be produced by mitosis, what should be the ploidy of the organism? Haploid. Haploid. Therefore, gametes have to be produced by mitosis. Alright? If the organism is haploid, then its life cycle is gametophyte is the dominant phase. Gametophyte is the dominant phase. Alright? Life cycle has to be certainly haplontic partially. So haplontic life cycle. Haplodiploontic, diplohaplontic, haplontic, diplontic. These are the 4 life cycles. What is haplodiploontic? Gametophyte is dominant, sporophyte is dependent. Gametophyte is dominant, sporophyte is dependent. So sporophyte can give rise to zygote production, no? Gamete production there. Then from gamete onwards, the gametophytic life cycle begins. So gametes are produced by meiosis only there also. Zygote will not undergo meiosis even when it is haplodiploontic. Gametes are produced by the diplontic cells or diploid cells by meiosis. The phase gametophytes are continuing there. So in these 4, Fucus, Funaria, Chlamydomonas and Marchantia, haplontic life cycle is in Chlamydomonas. Fucus, diplontic, Fucus is diplontic. Fucus is diplontic. Funaria and Marchantia both are bryophytes. Bryophytes have what kind of life cycle? Haplodiplontic. Pteridophytes have diplohaplontic. Gymnosperms and angiosperms have angiosperms diplontic. They have diplontic life cycle where only the gametes are the haplontic representatives. Okay. So Chlamydomonas. Life cycles of Ectocarpus and Fucus respectively are, see continuity. Fucus, what life cycle we saw now? Diplontic life cycle. Ectocarpus and Fucus respectively are haplodiplontic and diplontic. With reference to factors affecting the rate of photosynthesis, which of the following statements is not correct? Atmospheric carbon dioxide concentration up to 0.05% can enhance carbon dioxide fixation rate. The law of limiting factors. Who gave the law of limiting factors? Blackman. Blackman's law of limiting factor states what? Whatever is the factor which is available in deficiency, that is the one which will be the factor that limits the process. Carbon dioxide availability is how much? What is the percentage of carbon dioxide available in the 0.03% is carbon dioxide availability. If you increase carbon dioxide concentration, photosynthetic fixation of carbon dioxide also increases. But if you keep on increasing it, will the photosynthetic fixation continue to increase? It will fizzle out. So statement 1 is correct. Statement 2, C3 plants respond to higher temperatures with enhanced photosynthesis, while C4 plants have much lower temperature optimum. Temperature optimum is higher for C4 plants and not C3 plants. So this statement is wrong. Tomato is a greenhouse crop that can be grown in carbon dioxide enriched atmosphere for higher yield. That is also correct. Light saturation for carbon dioxide fixation occurs at 10% of full sunlight. Yes. Which of the following cell organelles is responsible for extracting energy from carbohydrates to form ATP? Which organelle will extract energy from carbohydrates to form ATP? Ribosome, chloroplast, mitochondria, lysosome. Mitochondria. Which of the following statements is correct? See, the statements are all related to descending and ascending loop of Henle and absorption of water and electrolytes. Don't look at the options. Tell me, the loop of Henle, there is exclusive absorption, exclusive permeability. If it is exclusive to, if it is permeable to water, it is not permeable to electrolytes and vice versa. Now you have to tell me which part of the loop of Henle is permeable to water and which one is permeable to electrolytes. Descending loop is permeable to water, ascending loop is permeable to electrolytes. As a corollary to that, by default it is not permeable to the other. Now look at the options. First option, descending limb impermeable to water, wrong. Ascending limb permeable to water, wrong. Descending limb permeable to electrolytes, wrong. Ascending limb impermeable to water, correct. That's one. A baby boy aged 2 years is admitted to play school, which is wrong, not the right age to admit the child to a school, and passes through a dental checkup. Alright? The dentist observed that the boy head is no longer fitting into the socket firmly. It becomes loosened and it falls off. It is not falling off because the permanent teeth that is coming from underneath is pushing it. Alright. Some people tell that because permanent teeth that is coming out is pushing out the temporary teeth. It is not the CM's chair, no? Somebody pushes the existing CM and comes and sits there. Okay. Premolars are absent 2102 by 2102. What is the dental formula for permanent teeth? 2123 by 2123. Each quadrant has 8 teeth in permanent teeth. Each quadrant has 5 teeth in deciduous teeth or temporary teeth. Which one of the following statement is not valid for aerosols? What are aerosols? What are aerosols? Suspended particles that you find in air. Aerosols in this context are pollutants. Aerosols are pollutants that are suspended in the air that we breathe in the atmosphere. They alter rainfall and monsoon patterns. Is it true? Do they alter the monsoon patterns and rainfall? Aerosols do, because they have the potential to change the wind pattern, the light absorption, all that. They cause increased agricultural productivity. No. They have a negative impact on agricultural land. Yes, because the pollutants, when they come down to the land with rain, they can cause negative impact on the agriculture. They are harmful to human health. Definitely. There is one person, I forgot his name, my wife told about him. He apparently is monitoring every bit of his health, including the DNA, methylation and all he monitors. He is trying to be eternal, it seems. You know him? You know him? I don't remember his name. I came to know about this person very recently. He had come to India and he had come to Delhi for an interview. Conditions were created for him so that he gives the interview in conditions which he is comfortable in. Midway during the interview, he just left the interview telling that the air is not within the parameter of acceptability for me, and he left. He would have died probably if he had gone on the streets of Delhi. For interview, he was brought in that very controlled environment. He came, and here also he was sitting in that room where the entire thing was controlled, air quality was monitored. And then he tells that this is not good enough for me to sit here, I'll go. So much of criticism happened. But he gave insight, he made people look into something which we are ignoring. Water, we can have filters. Water filters and all you have, and you can purify water and drink. Food, you are growing organic food, you can cultivate food within your own home and eat. Air, all these pollutants, aerosols and these things are actually carcinogens. They all are potential carcinogens. We are constantly inhaling, and if some chemical is on our skin, it takes a longer time for it to enter the bloodstream. What we eat takes time to enter the bloodstream. The gaseous pollutants that we inhale gets into the bloodstream quickly because of the the alveoli and capillary barrier. Very thin, only 2 layers of cells. Diffusion happens and it gets into the bloodstream. Very dangerous. Okay. Lungs are made up of air-filled sacs, the alveoli. They do not collapse even after forceful expiration. Because, why? Why don't they collapse? Because of residual volume. Because of residual volume. Tell me the different volumes. I will tell you the condition, you tell me which volume it is. The air that I inhale or exhale during normal respiration. Tidal volume. What is the volume? Around 500 ml. The air that I can inhale to the maximum capacity after my normal inhalation. Normal inhalation and beyond that. Inspiratory, reserve, inspiratory, reserve, volume. After a normal exhalation, the maximum air that I can inhale, which includes tidal volume plus inspiratory reserve volume. Inspiratory capacity. What I can exhale after I do normal exhalation. Expiratory reserve volume. After a normal inhalation, the maximum I can exhale. Expiratory capacity. The air that I can inhale to the maximum after maximum exhalation. Maximum exhalation to maximum inhalation. That is the vital capacity. The reverse also. Maximum inhalation, from that point, if I exhale to the maximum, that is the vital capacity. No matter what I do, that volume which I cannot touch is the residual volume. Residual volume is more or less equal to the expiratory capacity, around 1000 to 1200 ml. A little more than the expiratory capacity also. Inspiratory reserve capacity, inspiratory reserve volume is higher. Alright, tidal volume is 500 ml. So if you get questions where you have to arrange in the increasing order of volume or decreasing order of volume, remember residual volume is slightly higher than expiratory reserve volume. But if the thing that you are supposed to put in is expiratory capacity, which one is higher? Residual volume or expiratory capacity? Capacity is higher, expiratory capacity is higher. Alright? Very slight difference between residual volume and expiratory reserve, where residual volume is slightly higher. But if you add tidal volume to the expiratory capacity and make it expiratory reserve volume, then that is higher than residual volume. So pay attention to what terms are given when questions relate to arranging the volumes in increasing or decreasing order of the volume. Function of copper ions in copper releasing IUDs is, we have had extensive discussion on IUDs. Copper releasing IUDs release copper. IUDs are intrauterine contraceptive devices. Role of copper is, it will alter sperm motility. It will alter sperm motility, and it will also interfere with capacitation of sperms. So it will not do anything to ovulation. It will not prevent the endometrial development completely, though it will prevent it partially. It will reduce the endometrial thickening. So even if fertilization happens, implantation cannot happen effectively. So there will be a very early stage abortion. But even fertilization does not happen very often because copper will prevent motility of the sperms and capacitation. So sperms will not be able to reach the ovum to fertilize it. Even if some very active sperm does it, implantation cannot happen because the endometrium doesn't become as thick as it is required for implantation to happen properly. Alright? Next. Which of the following options best represents the enzyme composition of pancreatic juice? You want to go through or skip it? Skip it. We don't want any unwanted people in the room. Okay, this stage, no general knowledge, only specific syllabus related discussion. But unfortunate that digestion has been removed from the syllabus. I am not able to digest it. Okay. Asymptote in a logistic growth curve is obtained when? What is an asymptote? Asymptote is a limit towards which a function approaches. Asymptote is a limit. So infinity. Can I call infinity as an asymptote? You can never achieve infinity. No, you tend to move towards infinity. The ideal engine is, let's say, the asymptote or the limit to which the asymptote to which the engine can move towards is 100% efficiency. Asymptote is the limit that a function approaches but never reaches. So infinity is an asymptote in that sense. Alright? Asymptote in a logistic growth curve. This is population genetics. Two types of growth curve is noticed: exponential growth curve and logistic growth curve. Exponential growth curve and logistic growth curve. Exponential growth curve is not a practicable model because no population grows exponentially forever. Logical growth curve. It is called logistic growth curve because initially there is a slow increase and then there is an exponential growth for a while and then it eventually stabilizes. What is K, N, R, all of those? What is R? R is the intrinsic growth rate. What is K for a population? Carrying capacity. What is N? Size of the population. Now when the population is small, the growth rate will be, when the population is small or very less compared to the carrying capacity, growth rate will be higher. Population grows at a rapid rate when the population is small. Why? Resources are available. Resources are available. The population that is present to use the resources is lesser. So the reproductive rate increases, increasing the rate of growth. Rate of growth and the actual number of growth is different. Alright, rate of growth is for every person that is there in the population, what is the change? Number of people, when the big population is there, even if the rate of growth is lesser, the number of people being added will be more compared to a smaller sized population with a higher rate. Example, a population of 100 people grows at a rate of 10% annually. What will be the total number of people added to this population at the end of 1 year? Huh? A population of 100 people grows at a rate of 10% for this 1 year, coming 1 year. What is the total number of people added to this population at the end of 1 year? 10. A population of 1000 people grows at a rate of 5% annually for this coming year. What is the total number of people added to this population at the end of the year? 50. So when you look at absolute numbers, population 2 added 50 people, population 1 added 10 people. Which population is growing at a higher rate? The absolute numbers will not tell you unless you have the baseline value from which the number has changed. So population 1 has a growth rate of 10%, population 2 has a growth rate of 5%. When it comes to absolute numbers, population 2 has more numbers, number of individuals being added, population 1 has less number of people being added. Growth rate is a big indicator of demographic change than the actual number of people. So a population of a particular group of people might be 5% of the total population. The population of another set of people might be 95%. But population A, which is 5% of the total population, grows at a rate of 20% annually. Population B grows at a rate of 5% annually. When you look at absolute numbers, population B will always continue to have bigger number. But over a period of time, you take a longer time frame where population A is growing at a rate of 20%, population B is growing at a rate of 5%, the proportion will keep increasing for this population and the proportion will keep decreasing for this population. Eventually, when a time comes where the population of this group is sizable, this group becomes dominant on this group. So this happens in nature. A population that is growing faster at a higher rate is more likely to replace the population that grows at a lower rate, irrespective of the numbers, irrespective of the absolute numbers. So in demographic studies, it is important to understand the growth rate and not just the absolute numbers. Alright? So it has greater significance globally. Cultural transformation. America had native population in higher numbers earlier. A lot of migrants moved in. The rate of growth of population for the migrants is much higher than the native population. So over a period of time, the number of people who are originally migrants will be more than the number of people who are originally the native inhabitants. Power is always number driven, and power will always strengthen those who get power. So cut there, go after 100 years, the entire demography of the population is changed. Somebody existing at this time frame will feel this has been the composition of this population forever, unless they study history and see how the population has changed, demography has changed. Anyways, look at this question. Asymptote in the logistic growth curve is obtained. Asymptote is what? Reaching the limit. Alright? K is equal to N. K carrying capacity is equal to the population, which can never happen. The population will never reach the carrying capacity. K is greater than N, K is lesser than N, carrying capacity cannot be more than the number of people cannot be lesser than the number of people that is present. So growth will start slowing down when the population reaches its carrying capacity, and the intrinsic growth rate approaches zero at asymptote. Asymptote is reaching the limit. So when the population is reaching the limit of carrying capacity, the growth rate will reach the zero level. That is the asymptote. The value of R approaches zero. So if growth rate, if R approaches zero, what does that mean to the population? What does that tell you about the population? The population is stable close to its carrying capacity. N is close to K when R is close to 0. You understand? Higher the intrinsic growth rate indicates that the actual population is far below the carrying capacity. Reducing growth rate indicates the population is moving towards the carrying capacity. You understand? So in any logistic growth curve, you have a lag phase, exponential phase, deceleration phase, and a stable phase. Those are the phases. The region of a biosphere reserve that is legally protected and where no human activity is allowed is known as core zone. Core zone, as you move out of the core zone, what do you find? Outside the core zone you have buffer zone, and outside of the buffer zone you have transition zone. So in a biosphere reserve, in a reserve forest, where do we go for tour? Where are we taken for a tour? In the transition zone. Cheating. We are just being taken around the transition zone to see some animals by luck, but most of the animals are deep in the core zone. You know, there is a national park, Bandipur National Park. It is in Karnataka. Me and my wife went there. We went there for a trip. Bandipur National Park is elephant reserve. It's an elephant reserve forest. We went on the safari, half an hour safari. We saw a lot of dogs, pigs, all that. Dogs, pigs, and two wild bison we saw, wild buffaloes, which is just a little bigger than our regular buffaloes that we find in our village. And fortunately, we saw some animal suddenly going into the bush, and we told ourselves that it is fox. We are still not sure that it was fox, but we want to believe it was fox. Otherwise, 500 rupees per person was the safari cost. That is our defense mechanism to not feel bad about the money spent for the safari. Okay. Capacitation occurs where? Epididymis, vas deferens, female reproductive tract, ureter testis. Capacitation occurs in the female reproductive tract. Which ion is necessary for capacitation? Mineral. Calcium is necessary for capacitation. Hepatic portal vein drains blood to the liver from the intestines. So whatever is absorbed from the small intestine goes into the lymphatic channels. From there it goes into the hepatic portal vein, and hepatic portal vein goes to the liver before it gets again joined together to form the inferior vena cava, goes and joins the inferior vena cava. Why this portal system is necessary is because whatever we have eaten, this is a major portal of entry for chemicals into our body. Our gut is the major portal of entry for chemicals. Liver is our detoxification factory. whatever is absorbed from the intestine when it goes to the liver before it goes to the heart and gets into circulation, increases the possibility of detoxification and reduces the risk of damage because of toxins which might have entered our body through our gut. So it's a beautiful mechanism to ensure that as much as possible the toxins have to be removed from the blood which is coming from the intestine. If it goes into circulation, every other part of the body gets exposed to all of these toxins, and the elimination of toxins will be slow. Chances of damage is high. Right? That is why, due to alcohol. See, alcohol causes liver damage first. Why? Ethanol, when it is absorbed, goes to the liver. Liver will start metabolizing it, converts it into alcohol dehydrogenase, converts ethanol into methanol. Methanol is converted further into simpler metabolites, easy to eliminate by aldehyde dehydrogenase. But liver is getting exposed to this aldehyde. Aldehyde has a property to damage the lipids, membrane damaging action. So liver is constantly being attacked, damaged. Liver has great regeneration capacity, so it regenerates, continues to work. But if it is constantly being triggered by this aldehyde because of alcohol consumption, eventually liver will go into cirrhosis. So chronic alcoholics will develop liver cirrhosis. Liver cirrhosis is death of the liver tissue and accumulation of those masses. Fibrosis of the liver also happens. Okay. Have you read news or seen news about people dying because of consuming contaminated alcohol? Alcohol consumption leading to death of people. Alcohol consumption leading to blindness. Methyl alcohol. Methyl alcohol or even the aldehydes. High levels of aldehydes can lead to disruption of the membranes, detachment of the retina. It can cause retinal detachment, blindness. All of you have to stop alcohol consumption. Okay? Don't consume alcohol, not even social drinking. Useless. See, remember, what appears to be normal should not be accepted as normal. You need to have a moral framework of your own. You need to have a moral and ethical framework of your own, which is based on what is good for your life. What everybody else is doing may not necessarily be good for you. Everybody else in your office smokes. So is it okay to smoke? And justify it by telling that everybody is doing it, so what? It is not right. Everybody speaks in a crass language. It is very cool nowadays to talk in very crass and vulgar language. Is it okay? It degrades you as a person. It puts a very negative impression about you as a person. Because everybody speaks like that, is it okay? It is not okay. Some comedians are in big trouble now. Are you aware of the news? No? Somebody is aware. There is a show called India's Got Latent. Samay Raina. First of all, I did not like that show. Very vulgar. People talk all sorts of nonsense. And suddenly in that show, one more YouTuber called Ranveer Alabadia. Beer Biceps, some channel he has, very popular podcaster. He has come, he has made some really bad comment that has snowballed into a major controversy, and that show has been shut down. Cases have been filed across the country on them for passing some really crass comment. They are likely to be arrested for obscenity and vulgarity. Think what they might have told on the show as comedy. I will not tell what it is. You go and see it on 6th of May. Alright. So what is okay socially is not necessarily okay personally. You should have your own boundaries. Don't let yourself get degraded because everybody is in a degraded state. Sexually transmitted disease, causative agent. Again, match the following. What is our rule? Catch hold of one which you are absolutely sure of. Which one do you want to catch hold of now? AIDS, HIV, D1. Okay. 2, 3, 4 has it. Then catch one more. Come on, quickly tell one more. Huh? Gonorrhea, Neisseria gonorrhoeae. A2. Okay, done over. Only option 4 has it right. Gonorrhea, Neisseria gonorrhoeae, syphilis, treponema pallidum, genital warts, human papilloma virus. AIDS, HIV. Mycorrhizae are an example of amensalism, antibiosis, mutualism, fungistasis. What is it? Mutualism. Mutualism is positive interaction. Give me the sign for mutualism. Plus, 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 plus. Both are benefited. What is the sign for symbiosis? Symbiosis. Symbiosis is also plus, plus. What is the sign for commensalism? It can be plus plus or plus 0. Both benefit or at least one benefits, there is no harm to anybody. Amensalism is the opposite of commensalism. It is minus 0 or minus minus. Either both are harmed or at least one is harmed, nobody is benefited. Alright? What is the difference between mutualism and commensalism then? How do you differentiate? Both are plus plus. I've told you, I've given you examples of 2 movies. No? Can you think of a movie where the hero and heroine don't get to live with each other, but they continue to carry on with their lives? कुछ कुछ होता है. Have you seen that movie? Shah Rukh Khan?
We watched it here, sir.
Huh?
We watched it here, sir.
Here only you watched it? Okay. Kuch Kuch Hota Hai is commensalism. They both like each other, they are both happy with each other. But even if they are separated, they continue to live. Tell me a movie where part of the uterus. Select the correct route for the passage of sperms in male frogs. Testis to cloaca is the route. So all options give that. Vasa efferentia comes from after testis. That is correct. Urogenital duct to cloaca because there is no separate genital and urinary tract. So it has to be urogenital tract and cloaca in the end. Only the second option gets eliminated by that. There is passage of the sperms through the kidney also in frogs. So testis, vasa efferentia, kidney, Bidder's canal, urogenital duct, cloaca. Frog's heart, when taken out of the body, continues to beat for some time. Select the best explanation. Frog is a poikilotherm. Is frog a poikilotherm? It is. Frog is a poikilotherm, but the reason for heart to beat even when you take it out is not because it is poikilotherm. Frog does not have coronary circulation. Frogs do have coronary circulation, that is not related to beating of the heart, it is related to circulation. Heart is myogenic in nature, that is true. Frog heart is myogenic. The impulses come from the muscle. They produce the impulse, they are not dependent on the nervous system. And also heart is auto-excitable. Auto-excitable means what? Intrinsic pacemaker activity. Is our heart auto-excitable? Is our heart auto-excitable? That means the pacemaker can produce impulse without nervous system activity. We have the vasomotor center. We have the vasomotor center in the medulla. Vasomotor center will send signal to the sinoatrial node, which will determine how much should the rate of impulse generation be. Impulses are produced in the heart, but the rate is controlled by this thing, vasomotor center. Alright. So C and D are correct. Heart is myogenic in frog and heart is auto-excitable in frog. See, even in frog, the pacemakers have nervous system connection to regulate the heart rate. So to that extent, we can consider our heart also to be auto-excitable because the pacemaker is within the heart. Nothing in the body is outside of nervous system regulation. So depending on the context in which the question is asked, you have to come to a conclusion. Important characteristic that hemichordates share with chordates is, do they have a nerve cord or notochord? Hemichordates do not have notochord. They do not have the nerve tube which forms the spinal cord. They do have the gill slits. Pharyngeal gill slits are present in hemichordates also. For in chordates, gill slits are for what? Respiration. In hemichordates, gill slits are used for filter feeding. Presence of plants arranged into well-defined vertical layers depending on their height can be seen best in, what do you call this? Vertically arranged? Stratification. Stratification is found predominantly in tropical rainforest. Stratification is more advanced in tropical rainforest. Myelin sheath is produced by, who produces myelin sheath? Schwann cells and oligodendrocytes. See, astrocytes, Schwann cells, oligodendrocytes, all of these are glial cells. The nervous system has neurons and glial cells. Glial cells are supporting cells. Different types of glial cells we have. Astrocytes form the blood-brain barrier. Blood-brain barrier is formed by astrocytes. Schwann cells produce myelin sheath. Schwann cells produce myelin sheath. Oligodendrocytes produces myelination again. Schwann cells and oligodendrocytes both are producing myelin sheath. Schwann cells produce myelin sheath for peripheral nervous system. Oligodendrocytes produce myelin sheath for the central nervous system, neurons. Then microglia. Microglial cells are the immune cells in the central nervous system. They are equivalent to the Kupffer cells of the liver. Kupffer cells in the liver are immune cells. Likewise, microglia are the phagocytic cells of the nervous system. Phagocytes of the nervous system are microglia. Ependymal cells are present in the, where are the ependymal cells present? What do they produce? Ventricles. They are present in the ventricles. What do they produce? Come on. Ependymal cells produce CSF, cerebrospinal fluid. Alright. So Schwann cells and oligodendrocytes produce myelin sheath. Schwann cells are for the peripheral nervous system myelination. For CNS myelination, oligodendrocytes. Astrocytes form the blood-brain barrier. Microglial cells are phagocytes, immunity. Ependymal cells will produce CSF, cerebrospinal fluid. GnRH, gonadotropin releasing hormone, a hypothalamic hormone needed in reproduction, acts on. See, releasing hormones act on anterior pituitary. Options with posterior pituitary have to be eliminated. 1 and 4 talk about anterior pituitary. So, stimulate secretion of LSH and FSH, FSH and LH and oxytocin. Which one is correct? FSH and LH. Oxytocin is produced by hypothalamus, stored in and released by posterior pituitary. You have to pay attention. Receptor sites for neurotransmitters are present on. Well, this is repetitive. Many times we have discussed this. Where do the receptors present? Where are the receptors present? Postsynaptic membrane. Hypersecretion of growth hormone in adults does not cause further increase in height because epiphyseal plates close after adolescence, bones lose their sensitivity to growth hormone in adults, muscle fibers do not grow in size after birth, growth hormone becomes inactive in adults. Epiphysis is the ends of the long bones. Epiphysis is the growing end of long bones. It is called, there is a plate of bone, there is a plate of cartilage you can consider. If I write the long bone here, one of the long bones. Now here, you have epiphyseal plate. This is bone, this is bone. This plate here will be cartilage. This plate here will be cartilage. This is the epiphysis. That is the epiphysis in long bones when you are growing. Eventually, after the growth is complete, these epiphyseal plates are replaced by bone. It will no longer be cartilage, you will replace it with bone. Growth is not possible. So all those advertisements you see where height grow, tall you, put out in bus stands, behind autos, where you have one person, Photoshop, elongated by Photoshop, with one giraffe next to him. All those advertisements are fake. Those products will have growth hormones analogs. Growth hormone analogs. People who take that will develop acromegaly. Height will not grow. Skin will become thick. Hand and foot will become thick. I don't have any acromegaly. Okay? This is normal growth hormone influence only. If you just look at my hand and feet, it looks like acromegaly only, but it is not. I don't get proper footwear. It's very broad, like a monster's foot. Okay. Yes. So don't take those supplements. Don't take those tablets or creams. Such amazing marketing items come. They say you should see this. Earlier we used to have those. When we did not have too much of internet and all, these advertisements would come on TV. TV, Sky Shop, such advertisements, half an hour slots. You have to look at those people speak. Like, fast forward. I can't even mimic it now. अभी 10 मिनट में वो खत्म होने वाला है, अभी आप तुरंत आप इसको ऑर्डर कीजिए, इस जो नंबर आपकी स्क्रीन पे दिख रहा है उस नंबर पे आप मैसेज कीजिए, कॉल कीजिए, अभी 10 मिनट में वो डिस्काउंट खत्म होने वाला है. Have you seen that? Oh my God, you will feel like we'll do something. We have to pay that now. Let me do anything now. TV Sky Shop. So we used to get this advertisement also. So they will show people with who are short statured, fully depressed, not feeling energy to carry on with their normal life. Suddenly they will get this cream. They will start applying it on their soles. How do you want to grow? What will you do? You will apply this cream on your sole when you sleep. So while you sleep, your body is growing. And within 6 months, you become a giraffe. Nonsense. No regulation. You can sell anything in our country. All of you, please create some real products, okay? There is market for any product. But please create real products and sell it and make money. Earn money. There is difference. to earn money and to make money. Which cells of crypts of liver cone secrete antibacterial lysosome? What we should do now for this question? Skip. Because pancreas, digestion. Adult human RBCs are enucleate. Which of the following statements are the most, is or are most appropriate explanation for this feature? Why is it enucleate? They do not need to reproduce. They are somatic cells. They do not metabolize. Metabolism is there. They are somatic cells, no doubt, but somatic cells also reproduce. So that is a wrong statement. They do not need to reproduce. Who are you to decide? So there is, RBCs are being produced. So there is a need for RBC production. RBC production is also happening because of reproduction in its precursor cells. They need not reproduce because all the space available is used for oxygen transport. Okay. Alright, 160. So we could do 70 questions successfully. It will increase. Alright, we will break for lunch now. And yeah.